Here are 5 USMLE-style questions based on frame-shift mutations, splice site mutations, slipped strand mispairing, and the effects of coding DNA mutations. 1. Frame-shift mutation. Question. A 4-year-old child presents with progressive muscle weakness and elevated serum creatine kinase levels. Genetic testing reveals a mutation in the dystrophin gene that leads to the insertion of a nucleotide, causing a frame-shift mutation. Which of the following is most likely to occur due to this frame-shift mutation? A. A shorter protein with a truncated amino acid sequence and disrupted function. B. A longer protein with an additional functional domain. C. No change in the protein's amino acid sequence. D. Missense mutation resulting in the substitution of one amino acid. E. Silent mutation with no change in the protein structure. Answer: A. A shorter protein with a truncated amino acid sequence and disrupted function. Explanation: a frame-shift mutation occurs when nucleotides are inserted or deleted in numbers not divisible by 3, causing a misreading of the codons downstream of the mutation. This typically results in a truncated or altered protein, as seen in Duchenne muscular dystrophy, where the dystrophin protein is shortened and non-functional. 2. Splice site mutation. A patient with a history of anemia and spleen enlargement is diagnosed with a rare form of beta thalassemia due to a splice site mutation. Which of the following is most likely to result from this mutation? A. The intron is retained in the mRNA, disrupting proper splicing and leading to abnormal protein function. B. A frame-shift mutation causes a premature stop codon, producing a truncated protein. C. A silent mutation leads to no change in the protein sequence. D. A missense mutation results in the substitution of a single amino acid, changing protein structure. E. The mRNA is degraded due to an error in the spliceosome. Answer: the intron is retained in the mRNA, disrupting proper splicing and leading to abnormal protein function. Explanation: a splice site mutation causes improper splicing of pre-mRNA, which can result in the retention of introns in the final mRNA. This leads to the production of a dysfunctional protein. In beta thalassemia, such mutations can lead to reduced or absent hemoglobin production, causing anemia. Number 3. Slip strand mispairing. Question: a 45-year-old man with a family history of neurodegenerative disease develops symptoms of cognitive decline and choreiform movements. Genetic testing reveals an expanded CAG repeat in the 8 gene. What is the mechanism underlying this expansion, and how does it lead to anticipation in subsequent generations? A. Slipped strand mispairing during DNA replication, leading to the insertion of additional repeats and anticipation across generations. B. Frame-shift mutation causing a misreading of codons and premature termination. C. Splice site mutation causing impaired RNA splicing and protein dysfunction. D. Base substitution leading to the production of a misfolded protein. E. Silent mutation leading to no change in the protein sequence. Answer: A. Slip strand mispairing during DNA replication, leading to the insertion of additional repeats and anticipation across generations. Explanation: slipped strand mispairing occurs during DNA replication, particularly in regions with repetitive nucleotide sequences, such as the CAG repeat in Huntington disease. This leads to the insertion of extra repeats, resulting in an expanded polyglutamine tract. The phenomenon of anticipation occurs when the number of repeats increases in subsequent generations, leading to earlier onset and more severe symptoms. Number 4. Missense mutation in Huntington disease. Question: in a patient with Huntington disease, a CAG repeat expansion leads to the substitution of a glutamine residue in the protein's structure. What type of mutation is responsible for this substitution? A. Silent mutation. B. Missense mutation. C. Nonsense mutation. D. Frame-shift mutation. E. Splice site mutation. Answer: B. Missense mutation. Explanation: a missense mutation is a change in a single nucleotide that results in the substitution of one amino acid for another. In Huntington disease, the CAG repeat expansion leads to the incorporation of additional glutamine residues into the protein, altering its structure and function. 5. Frame-shift mutation in cystic fibrosis. Question: a 3-year-old child is diagnosed with cystic fibrosis after screening reveals an autosomal recessive mutation in the CFTR gene. The mutation is caused by a deletion of 3 nucleotides. Which of the following would most likely occur due to this type of mutation? A. A non-functional protein due to a truncated amino acid sequence. B. A longer protein with an additional functional domain. C. A silent mutation leading to no change in the amino acid sequence. D. A frameshift mutation resulting in incorrect protein folding and dysfunction. E. A missense mutation leading to the substitution of one amino acid. D. A frameshift mutation resulting in incorrect protein folding and dysfunction. Explanation: a frameshift mutation typically occurs due to the deletion or insertion of nucleotides not divisible by 3, causing a misreading of all subsequent codons. In cystic fibrosis, a deletion of 3 nucleotides, such as delta F508, removes phenylalanine from the protein, leading to a dysfunctional CFTR protein. Although this specific case involves a deletion of 3 nucleotides, it results in defective protein folding and function, characteristic of cystic fibrosis. These questions cover the various mutation types,